USC. So how these biosimilars are available in USC. Then later on moving on the European Union. Then of course I'll be talking about the current status in India. Then of course the major one is the biosimilar development process. Though it's a very big process, but I'll be talking about this process in a short. And later on I'll be talking about different challenges during the biosimilar development process. So there are a lot of challenges are there. So how these challenges may be overcome. So these I'll be talking about the challenges in the seminar. And the safety consideration, of course, the biosimilars are safe already, but how the safety or the efficacy that has been done for the biosimilars and any other issues with the safety. So that I'll be talking about. Then I'll be concluding the webinar with the future perspectives. So dear attendees, just I want to make you understand the meaning of biosimilars. So the first question that will be appearing in your mind is the meaning of biosimilars. What is biosimilar? So biosimilars are the biological product that are very similar and cheaper than the biological drugs. So here there are two things we call about biosimilars, and other one is the biological product. So both are biological product only, in fact. So the biological product which was already available in the market, that we call as a reference product. So based on the reference product, another biological product is produced, that is called biosimilar. That means we have a standard, and based on the standard, we will be producing another biological product. So that product is known as the biosimilar product. So the reference product is the one which was already available in the market, that has been approved by the agencies in the sense. So there are different agencies throughout the world. In case of USA, of course, you must be knowing FDA, so which was approving agent for the biosimilars. And similarly in EU, European Union, there is a agency called EMA, European Medicines Agency. So these are the agencies, they approve the biosimilars. So that means the biosimilar is a biological product that should be approved by suitable agencies, and that is available in the market, so that we can take as a reference product. So regarding biosimilar, of course, as I told you, it is biosimilars or biologicals, what it may be. These are created in the living cells. So it may be the microorganisms or maybe any other cell in which the product means it's a protein product. So biological means it's a protein product which is to be created, means manufactured. That means that so these protein products that cannot be synthesized just like conventional drugs or another generics. That means these are not chemical drugs. See, these are the protein product, biological product, and make sure that there's a big difference between the conventional drugs and the protein product. So the conventional drugs we call generics also, and these can be exactly copied, whereas these biosimilars that cannot be exactly copied. So those matter I'll be talking in this slide. So biosimilars are officially approved versions of original innovator product and can be manufactured when the original product patent expires. So attendees, please note that there is a question here. The question is that how biosimilars that can be produced? Is there any condition? Something like, of course, there are a lot of issues with the biosimilar manufacturing. So biosimilar product may be produced based on the reference product when the patent of the biological product, that means the original product, reference product, expire. That means that you have a standard, the expiry of the standard. After that only it is possible to produce biosimilar, because we will be copying the standard. But as I told you, we cannot copy exactly. It's not possible to copy exactly. So why? What is the reason? That I'm going to tell you now. So a biosimilar is not regarded as a generic of a biological medicine. That's what I told you. The exact copy is not possible. So in case of small molecules, the exact copy is possible. But in case of the biological product, it is not at all possible. There are a lot of reasons. Of course, some reasons just I'm going to tell you. The reason is that because of the natural variability. So there is a natural variability. Because of that reason only, it is not possible to produce the exact copy of the biological product. So natural variability in the sense there are a lot of issues. That means the production. So manufacturing. So during manufacturing, there are a lot of the steps are there. So these are the complex manufacturing process. So because of that reason, that's one of the reason, in fact, that will not allow the exact copy of the molecular micro heterogeneity. So somehow some small changes will be there always, but still we can say these are similar to the reference product. Then we call those product as the biosimilar product. So attendees, sometimes you'll be having confusion between biosimilar and biological product. So as I told you, both are biological product only. So biological product is a product which is initially developed without having any standard. So this becomes the standard. So that is the biological product. So biological products are generally large and complex molecules. So comparatively, these are complex molecules than the chemical drugs. So these products are produced through biotechnological way. So with the use of biotechnological techniques in a living system such as microorganisms, we can use microorganisms for the production of biological product, or we can also use some cells. So generally it should be derived from animal cells. And because of these reasons, so complex molecules, because of that reason, it is very difficult to characterize the molecular structure of this protein. So whereas the small molecule that can be easily characterized, but these large molecule, complex molecule, protein product that will be having lot of difficulty in characterizing. So this is the difference between the simple or small molecules and the protein molecule. Of course, I will be talking about some the examples of biological products. So this is normally the question which will be asked by the generally the students community. So they may be asking, may be interested to ask what are the biological products available in the market. So in this case, I want to give you only two examples. Of course, lot of examples are available. But however, in this slide, just I want to highlight only two examples with the uses, so that the students will understand how much the biological product or maybe the biosimilars is useful in the disease conditions. So biological product. Maybe the therapeutic proteins. The example is filgrastim. So that is one biological product. And there are so many monoclonal antibodies, but here I am giving an example as adalimumab. So these two biological products just I want to highlight here. Dear attendees, please note that the second one, the adalimumab. So anything that is ending with the mab, just you can make sure. So anything that is ending with mab, that means it is a monoclonal antibodies. So students may be interested to ask the purpose or maybe the uses of the biological products. Of course, if you see the uses, then you'll be surprised that there will be a vast and many uses of this biological product. That means filgrastim injection that is used to treat neutropenia. So neutropenia where the body will have low white blood cells. So this happens due to the cancer therapy in a cancer patient. So cancer patient receives chemotherapy, and hello, Ratna is friend, can you hear us? Hello.
Yes, sir, audible, sir.
Audible. Okay. Some information coming in my slide. That is reason, right? So filgrastim injection that is used to treat neutropenia. That's why I told you the low white blood cells, which is a condition that is by the cancer patient because of chemotherapy. So that condition may be treated with the use of filgrastim injection. Then the next one is the adalimumab. This is a monoclonal antibody which is used to treat different kinds of arthritis, ranging from several arthritis, of course, the rheumatoid arthritis, where you will have the inflammation of the joint. So you will feel very difficult in walking. That means no physical activity is possible. In that case, this adalimumab is a monoclonal antibodies, means it's a biological product, so that can be used. So students, knowing about the biological product, of course, we should know what is a reference product. So I told you, so this reference product or maybe the biobetter, these are the different terminologies that is used along with the biosimilars. So how these may be linked with the biosimilars? So that is a question, and of course, these are the important the terminologies that is to be considered. And I'll be talking about the reference product. So I already told you about reference product means this is the biological product which is already available in the market. So that we take as a standard reference. Based on this, the another biosimilar product is produced. So a reference product is a single biological product which was already available in the market and was already available by different agencies. Agencies in the sense, in case of USA, it is FDA is the agency that approved the biological product, that means reference product, or it may be approved by any other country by their agency. In case of European Union, it is EMA, European Medicines Agency is the agency that approved. So we can take the approved biological product as a standard based on which the biosimilar product may be developed and compared. So we will be using the reference product for the comparison purposes only, and the biosimilar product which is proposed to be developed should be having similarity with the reference product. So this is to be noted. Of course, in a simple way, I can say reference product is a standard that we use. So a reference product is already approved in the different agencies. And of course, how it is approved? Because it is having full safety and effectiveness data. That means it is already proved to be effective. It is already proved to be safe. That means the biosimilar product that is proposed to be developed, that should also have the safety profile and effectiveness data. And of course, it's a comparison between the similar product, biosimilar product with the reference product. And of course, a proposed biosimilar product is compared to and evaluated against a reference product to ensure that the product is highly similar and has no clinically meaningful differences. So sometimes you may be having some confusion. So already reference products are available in the market, and another company may be producing biosimilar product. So why? What is the reason? Okay. So there are a lot of reasons that I'm going to tell you in the future slide. But here I want to make one thing, that when making comparison between the reference product and the biosimilar product, make sure that the biosimilar product is safe and effective, and that should be highly similar to the reference product, and there should not be any meaningful differences clinically. That means the biosimilar product is not the substandard product. So I want to highlight here that the biosimilar product is not substandard product of the reference product that is having the equivalent activity. But we cannot do the bioequivalent studies in this case, but we can say the totality of the evidence of the biosimilar product is very close to the reference product, so that there is no big differences. If there is no big differences, there is no difference in the the activity or maybe the safety, and we can say there is no clinically meaningful differences. So when you make such a kind of product, then we can say that it is biosimilar to the reference product. Okay. So dear attendees, there is another the terminology generally used is the interchangeable product. So this also a biosimilar product. So it's a biological product only, it's a biosimilar product. So interchangeable. So we can change, right? So this is a product that can be expected to produce same clinical result as the reference product. Of course, generally all the biosimilar product should have same kind of activity, safety data, same thing only. But here we call as a interchangeable product. So where is the difference? So the difference is that, so this is a biosimilar product which is approved by a different agency as an interchangeable product, so that the pharmacist may substitute only pharmacist, so that the doctor intervention is not required. Only pharmacist, he can substitute the interchangeable product to the right reference product. So it is a duty of the pharmacist now to substitute a reference product to the patient without the intervention from the healthcare provider. Generally we call him as a doctor. So that is the one which is available generally in the USA. So they use this term interchangeable product, and that should be approved accordingly. So students, there's another the term which is called biobetter. So sometimes this leads to some confusion. Why that leads to confusion, that I'm going to tell you now. So before that, I'll tell you the meaning of biobetter. So biobetter refers to a protein product that is in same class as an existing biopharmaceutical. So it's a biological product. It is not identical. It is improved over the original product. So you can understand now, biobetter. So it's a biological product. It's better than the biological product. That means what? It's a biosimilar product. When compared with the reference product, it is having good activity, it may be, or maybe it has some improved property. You can see in general, it may be good activity or faster action, anything. That means it is having some better activity than the reference product. So if it is having better activity than the reference product, this product cannot be considered as a biosimilar product. But initially it was developed to get a biosimilar product, but later on it has been seen that it is better than the biological reference product. That means it's called biobetter. So this biobetter is not the biosimilar product now. Okay, so there will be a confusion again here. So if it is biobetter, how it is better? So I tell you, biobetter will show improvement in one or more attributes, means qualities or properties over the original biological drug. That means when compared with the reference drug, it is having some better property, good property. So one example I can give here, that the activity, I can say the property, it may be having the better side effect profile, means it shows less side effect when compared with the original drug, or it may have some faster action than the original drug, or it may require lower dosing when compared with the original drug. So any one quality, if it is superior than the original product, then this is called biobetter. So biobetter also considered to have different active component when compared with the original product. That means I told you, so biobetter initially developed to get a biosimilar product only, but later on it has been noted that it is better than the biological product, reference product, then we call as a biobetter, because of some reason, some good properties that is exhibited by the biosimilar product. Now it is not biosimilar, we call biobetter. Okay, so biobetter is another biological product now. We cannot say it's a biobetter, biobetter as a biosimilar. It is another biological product, so original biological product, but it is based on the another reference product. So having this idea, I'll give you one example of biobetter, that there's a monoclonal antibody which is known as obinutuzumab. So maybe a little bit difficult to pronounce, obinutuzumab. So this is called a biobetter of rituximab. Rituximab means it's a rituximab. So this is another monoclonal antibody. The trade name is rituximab. So when compared with this rituximab, obinutuzumab is better. How it is better? Because it is less immunogenic and it is more cytotoxic when compared with the rituximab. So that it is called biobetter. So dear attendees, please note that sometimes the researchers, they don't accept the word biobetter, because always you are comparing one product with another product. Where you say one product is superior, another product is inferior. So this comparison generally is not acceptable by many of the researchers, and sometimes from the the patient point of view, of course, we want to have a better product, then we call as a biobetter. So this is a confusion among the researchers and the different stakeholders, whether to use biobetter word or not. But however, sometimes both are equally active. Of course, in case of different disease, between one point it may be better, in another point the another one may be weaker, but sometimes in another condition both are maybe equally active. So that the researchers, they don't want to use the name biobetter. So however, these are available. Since I want to make you to understand the meaning of biobetter, just I'm giving you this. So attendees, is it audible? Hello?
Yes, sir, you can proceed.
Okay. My voice is clear to you?
It is clear, sir.
Okay, please. So, dear attendees, I'll be moving on to the next, topic of biosimilar is the benefits. So, benefits nothing but advantages. So, there are a lot of benefits, or we can say advantages of using the biosimilars. So, how that is useful? What are the advantages of these biosimilars? So, that is a question that will be maybe ringing in the minds of the attendees. So, initially, the biosimilars were developed because of reduction in the cost when compared with the original biological product. So the cost reduction by competition. So this is the main agenda of developing the biosimilar product. And later on, there are a lot of other advantages also using this biological product. So just I will summarize. Biological medicines are costly in general and consumes a large portion of public and private drug spending. So when you take about the private hospitals or maybe the government hospitals to treat a patient, when you use the biological product, of course, biological products are very costly and you have to spend lot of money only for this. So that means we have to have some alternate. So the alternate is the biosimilars. So in most cases, biosimilars are less costly than the innovator biological drug. So innovator biological drug in the sense original one, that means reference drug. Why it is so? Why biosimilars are less costly? So there are reasons. Why? Because these biosimilars require fewer clinical trials. That means we need to have only fewer clinical trials only. So we need not do more clinical trials. So that is one reason. So because of this reason, we will be saving a lot of money in doing the clinical trials. So you can sell biosimilar and cheap in the market. So that's one. The second is that the cost of discovery and initial development of the drug is not at all needed. So for reference drug, you have to have a discovery and development cost. So the company spend lot of money for the drug discovery and development. But in case of biosimilars, already we have a reference product. Based on the reference product, we are producing the biosimilar so that the initial cost of discovery, the initial development, which is not at all required. So we'll be saving lot of money in this case also. So these two are the main reasons. There is another reason. The additional competition might help explain the cost differences. Suppose a company, they produce a biological product and of course spending a lot of money, and they want to take back the money, and they make a price in the market which will be very high generally. So they becomes the monopoly of that particular drug. If biosimilar is available to that particular drug, then there'll be a cost which will be reduced so much. To give you one example, before that there's a one word. Biosimilar medicines offer potential economic benefit to the healthcare system. So the same thing only, because when you produce biosimilar, then the cost of the medicines come down, becomes cheaper, so that will offer a potential economic benefit to the healthcare system. The government spend lot of money to the healthcare system. So if the product is available cheap, the medicines available cheap, then the healthcare system, the burden will be less. Okay. So in this case, just I want to give you one example of biosimilar product, how the price has been reduced. In Europe, the introduction of biosimilars have reduced the prices of the biologicals. The example is that in October 2018, 80% price reduction on the monoclonal antibody Humira. So Humira is a trade name that contains adalimumab. That is a monoclonal antibody. I already told you the uses also. So after the introduction of several biosimilars. So that means what, in Europe there are so many biosimilar products that contain adalimumab that has been introduced, so that the reference product adalimumab sold in the trade name of Humira, that company has to reduce its price up to 80%. That means if it was 100 rupees, now it has to sell for 20 rupees only. So that much of price reduction is possible because of the introduction of biosimilars. And having multiple biosimilars in the market will break the reference drug monopoly and help bring down the cost. So this already told you. So the cost reduction is possible because of the competition only. The competition means introduction of several biosimilars, and not by one company, by several companies, same product. if it is introduced, then the cost of the biosimilar product will be reduced. So this was the main aim in developing the biosimilar product. Okay. Then I will talk about the other benefits or advantages. These advantages are, of course, supportive only. The main advantage is that the cost reduction. Now the competition and innovation. So there are different companies, several companies involved in the biosimilar production. So because of that, there's a competition. Because of the competition, there is innovation. So the company may innovate some kind of biosimilar product. So new discovery is also possible. And enhanced patient accessibility. So this is also an important point to be noted, advantage or benefits to be noted. So biosimilar medicines may improve patient access to the biologicals. That means there are a lot of biological drugs available in the market. So biosimilar is also biological medicine only. If it's available in the market, then the patient will have a easy access to the biologicals. So because of that reason, there is a possibility of treatment options. This is also very important for patients who have serious health conditions. So there's a easy access to the patient. That's one thing. And second is treatment option is also possible. So these also, the further advantages of this biosimilar development. Then the biosimilar development that allows some healthcare system to use this innovative treatment for the patient. So some healthcare system, they follow new and innovative treatments for the patient. So this also for the the benefits in terms of the patient's view. Then the incentive for innovation. Of course, if we want to innovate something, we have to have a fund. So the government should fund you, then only is possible. In this case, the funding is also possible because of the biosimilar development. The pharmaceutical companies would be encouraged to invest more into the cutting edge R&D sector to foster innovation. So for innovation, for new drug discovery, it is required to have the funding, and funding also is possible because you are going to develop a new biosimilar product. So funding also is seen. And some government, they release the funding to the healthcare system that can keep pace with the growing healthcare needs, and funding new generations of the innovative treatments brought about by advances in the medical science. Because of the improvement in the medical science, advances in the medical science, so the funding agency also fund to innovate new treatment methods or maybe new biosimilars, etc. So these are the different benefits I can say with the development of the biosimilars. So here I'll come to the another topic which is called biosimilars. Why now? So dear attendees, you must be knowing biosimilars are recently available. The first biosimilar was introduced in the European Union. It was in 2006. So after 2006 only the people throughout the world, they talk about the biosimilar. And now many people, of course, everywhere you can see there's a talk on biosimilar. Why now? What is the reason the people are talking more and more about the biosimilars now? So that is the question generally. So here that relate to the patent, right? So patent on some brands of biological medicines have run out or reaching at the end of their term, which is why different pharmaceutical companies are making biosimilars of these medicines. So, dear attendees, please recall my earlier talk that the patent is playing an important role. So, once the patent of a original product expires, then only the company is able to make the biosimilar product. That means that time has come now. Patent of some biological medicines have run out already, so expired already, or some biological product patent is going to expire. So that is the reason many pharmaceutical companies are making biosimilar product. That means what? Once you make a biosimilar product, you can make money. So the company aim to make money. So this is one of the way. See, in that case, they have to spend a lot of money. How it is possible? Once the patent of the particular reference product expires, then only it is possible to produce biosimilar. And now is the time where the company may produce biosimilars and make some money. So that is only one reason. When patent expires on branded biological product, different pharmaceutical companies are allowed to make the biosimilar products. So this I already told you. So an approved biosimilars are expected to have the same safety and beneficial effect in patients as an existing biological medicine. So this also I told you. So the biological product and biosimilar product that will be having same activity, same safety profiles. So there is no substandard in this case. So that is the reason. And not only that, biosimilar products are also approved by the same regulatory authorities and are manufactured following the same high standard quality standards as for existing biological medicine. So that means what there is no difference at all. Whatever the biological product that is to be manufactured with the high standard, high quality standard, the same standard to be followed even for the biological means biosimilar product, so that you should not have any confusion with respect to the substandard product or low quality product. So we produce biosimilar product. Once the patent, that should expire after that only. So here to support you, I'm giving the biologics having already expired or nearing primary patent expiry in the US. So this is the US patent expiry or the date or year which is given here. There are I think more than 12 product that the patent got already expired up to 2019, and only few are available in US patent I'm talking about. And there is a one product, the expiry year which is given as 2028. Please note that. So the name of the product is Etanercept. So just you can make a note of this, because I'm going to talk about this in the next few slides. After a few slides. So here filgrastim is there, heparin alpha, pegfilgrastim, adalimumab, rituximab. Then there are so many things are there. So these are different, the monoclonal antibodies, or you can say generally biological product, or you can say your protein product, the expiry, patent expiry in the US. So that I'm going to means that I showed you. Right. So having this, I want to give you some current status of the biosimilars. Of course, I selected 3 regions. One region is the European Union. The second is the USA, because these two are the, you can say European countries. European Union means there are 27 countries. They formed an union we call European Union. So this agency and US agency, they are the one, they are pioneer, and they have very good standards, strict standard. Based on these standards only, the other, the countries, they produce the biosimilars. So we can have these two as a standard. Of course, we should not forget our country. In India, what is the current status? That I'm going to talk in this seminar. So the first biological product, biosimilar product. The first biosimilar product which was introduced in the European country, that is the European Union, it was in 2006. So the name of the biosimilar is Somotropin. So there's a name of the biosimilar. Okay, so this Omnitrope is the trade name, brand name, you can see, right? So dear attendees, just make a note. This is the first product which was approved in the European Union. So this is the first one throughout the world. Okay, so after 9 years later, then the USA introduced the first similar in USA. So up to now, maybe this is the, what to call this reference. Maybe I have collected some 2 months back, maybe up to the end of April. There were 64 biosimilar products are available in the European Medicine Agency. That means the European Medicines Agency approved or recommended 64 biosimilar product. So this I am talking about up to April, maybe at the end. I don't know, May and June, I don't know. These 2 months I did not check it. So maybe available, may not be available, I'm not sure. But however, up to the end of the April 2020, we have 64 biosimilar products which was approved in the European Medicines Agency. So these are classified into different categories. Of course, depending upon the agency, there are different categories of the, the biosimilar products. So as per the EMA, whatever is available, that was categorized under 8 different categories. The human growth hormone, the example is Somotropin. Granulocyte colony stimulating factor, that is filgrastim. So I told you about filgrastim, and please note that attendees, please note that there is a suffix AAFI. Are you able to see this one? I tell you why this one. Why this suffix is there after the filgrastim. So suffix always say that it's a biosimilar product. Suffix which is there along with the another name, then we can say that this is a biosimilar product. So that is we can say nomenclature recently that is introduced. Okay. So similarly, we have another one. Erythropoiesis stimulating agent, E14 alpha. Again, there's a suffix. Then insulin is there. Sometimes the cost of insulin is increasing, where all other biosimilar products are, the cost is decreasing. The insulin is increasing because of some reasons. Of course, I don't want to give you big reason for this insulin, but there are reasons. Then you have follicle stimulating hormone, pholitropin alpha. It is in the brain of overlay. Parathyroid hormone. And we have a tumor necrosis factor inhibitor. So there is infliximab. So with the suffix, then we have a lot of monoclonal antibodies. Even anything just ending with the mab, that is also monoclonal antibodies. There are several antibodies which is given here, and these are the biosimilars. So CT, P10, these are biosimilars. So these biosimilars are produced based on rituximab. That means it should be given as rituximab, some suffix should be given. So these are the different biosimilar products are available in the European Medicines Agency. So dear attendees, have a very big the list, and of course it is in the form of table. Just I want to tell you to make you understand how these approved biosimilars are available in the EMA. So the first one, E14 alpha, of course, it is useful for different purposes like anemia, cancer, etc., and when it is authorized and which company that manufactures. So these are given here. And in this case, I just even highlight the third and fourth. Amjevita is a company, it's a product name, and Amsparity is a brand name, you can say. So these two produce adalimumab as a biosimilar product. And the first one, Amjevita, which is produced by the company Amgen, whereas the another one, Amsparity, that is producing, this is a brand name that contain adalimumab that is produced by Pfizer. That means different companies produce same biosimilar product and have different brand name. So that means what? There are several companies are interested in producing the biosimilar product. Because of that reason, the cost of biosimilar product becomes less. So here also you can have etanercept. So I already told you about etanercept. The biosimilar of etanercept is Benipali. So students, or we can say the attendees, please just recall, I already told you about etanercept. The patent of this in US is up to 2028, but already we have biological product, means the biosimilar product. It may be Benapali or maybe this one. It is already available. How it is possible? That's a question generally that should appear in your mind. Yes? Yes. So how it is possible? Because here the patent of this etanercept in EMA expired, whereas the same product in USA it is not expired, but in EMA, that is in European Union, it's expired. Because of that reason, the company producing the biosimilar product with the different brand name. So in this case, the Samsung Biops that produced one, and the other company, the Sandoz, also produced the biosimilar that is based on etanercept. So similarly, we have filgrastim and we have pegfilgrastim. It may be there. It's not coming. So you have pegfilgrastim. So students, there is a similarity, pegfilgrastim and filgrastim. So these are very close, in fact, but these are different biological product, and there are different biosimilar product. So filgrastim is a biological product, and pegfilgrastim, these are very close, but here PEG, that means it's a pegylated PEG, polyethylene glycol. So pegylated form of filgrastim is the different biological product, and these are having very similarity. And of course, these are different biological product. So this is just I want to tell you, because you should not be confusing with filgrastim and pegfilgrastim. So both are having very similarity, where the filgrastim is pegylated, that becomes pegfilgrastim. So is it audible? Ratanarisware? Hello.
Sir, you can proceed there.
Yes, sir.
Please, because just I want to check whether my voice is clear or is it audible. Everything, just want to check it. Nothing else. Okay.
No issue, sir.
Okay, please. Then now I'll come on to the status in USC. So as I told you, after the introduction of biosimilar in means European Medicine Agency, after 9 years, the USA that introduced the biosimilar product in USA. So that is called filgrastim S and DZ. So this S and DZ, I already told you, this is the way of making nomenclature for the biosimilar product by the USA, so that they have 4 suffixes. The suffix does not have any meaning, but simply they suffix it, and this is to be given during the approval of this medicine. So the USA FDA, they approved filgrastim biosimilar product with the name suffixed with S and DS, DZ. That means it is in the name of the trade name of brand name of Zarzio. So this was the first biosimilar product that was approved in the United States in 2015. So that means after 9 years from the first biosimilar product that was launched in EMA, after that the USA that they have introduced. So this is a product biosimilar to filgrastim. So this original product, the company of this original product is Neupogen. So the FDA consider a product biosimilar if it has same mechanism of action, route of administration, dosage form, and even strength of the biosimilar product should be same as that of the original product. Means everything should be same. If it's any deviation, that will not be considered as a biosimilar product. So the FDA regulatory conditions are very, very strict. That means the biosimilar product should possess same properties as that of the original biological product. So in this case also same. So that is the reason they have approved this Zarzio brand name of filgrastim. A biosimilar product must have the same clinical indications for use as the original product. So this already I told you in the earlier, but here also, even US also, they follow the same conditions. In this case, the original biological drug, the filgrastim, which is manufactured by the company Amgen. So Neupogen is the trade name, brand name. So the company is the Amgen. The biosimilar filgrastim S&DS, it is manufactured by Sandoz. Hence, there is an unusual suffix. I already told you the suffix that indicating that there is a biosimilar product. So this is to avoid the confusion between the original reference product and the biosimilar product. So they introduced new naming method, nomenclature. So that nomenclature, what it says, you suffix the original, means biosimilar product with the some suffix, some name, some 4 letters. This is the reason. So biosimilarity does not guarantee bioequivalence. So I already told you it's not possible to produce a bioequivalent between the original product and the biosimilar product. It's not guaranteed. We cannot give any guarantee of the bioequivalence. Hence, the filgrastim cannot be substituted for the filgrastim, that is the original product, without direct approval by the healthcare provider. That means what? Only the doctor, he can substitute. Otherwise, it is very, very difficult to substitute. So substitution is very, very difficult to make. Until otherwise, it should be approved as an interchangeable product. And of course, in this case, the adverse reactions, the safety issues, everything that should be approved. And there may be slight differences. That is not impossible. That is no problem. But however, that should show very, very similarity with the original drug. So dear attendees, I show you there are 19 products right now available in the USA market. In EMEA, there are 64, but in USA, only 19 products are available. Of course, maybe this is the literature up to the end of the April, and after that I don't think so. But however, maybe one or two may be also available. So only few available in the market, USA market, biosimilar products. So what is status in India? Of course, it is available. So biosimilar products are available in the India. So several products are approved by the different agencies in India. It's a good point. Of course, we have access to the biological products. Many products are available in the market. It's a good sign that. So the biological product in India are called means the biosimilar product. Biosimilar product in India, it is called similar biologicals. That means what? There's a difference in the naming. So when you say biosimilar in USA or in European Unions, so it's not a problem. But when you call biosimilar in India, it's wrong. We call as a similar biologics. So the Indian regulatory agencies, they call biosimilar as similar biologics. Okay, so these similar biologics in India is available in different categories like vaccines, monoclonal antibodies, insulin, and recombinant proteins. Of course, depending upon the country, there are different classifications. So in India, there are 4 classifications. Of course, maybe in the future there are more. There may be several other classifications also may be possible. But however, we have to understand that these are the biological products or biosimilar products available in the market. So there are many Indian pharmaceutical companies engaged in manufacturing and marketing of biosimilars. Yes, it is already available in the market. In Indian market, for biosimilars will be over 35 billion by 2030. So maybe another 10 years will have a very good market potential for the biosimilars. So if you ask me what are the pharmaceutical companies that are showing interest in developing the biosimilars, there are we call multinational companies generally we call and or the Indian companies, they have the Indian companies in collaboration with the multinational company, they produce and they market biosimilars, namely Biocon. We have Glenmark Pharmaceuticals, Cipla, Lupin, Aurobindo. Of course, there are so many others also, Zydus Wellness. So these are the multinational companies. They focus on the biosimilar, drug development and also marketing both. So what is the regulatory agency? They control the biosimilars in India. So that is a normal question that arises. Of course, it is the CDSCO. To address the issues and challenges associated with the development of similar biologicals, Central Drug Standard Control Organization, in short it is called CDSCO. So along with, means in collaboration with the Department of Biotechnology, so they have developed guidelines for biosimilars. Of course, I told you in India, we should not call biosimilars, we call similar biologicals. So that's what guidelines on similar biologics, regulatory requirements for marketing authorization in India. So this is the guidelines available in India. So based on this guideline, it is possible to produce, or we can say manufacture, development, marketing of the biosimilar product. So this was initially introduced in 2012 and later on it was revised in 2016. So these guidelines give a lot of information. The regulation of manufacturing process as well as quality, safety, efficacy. Generally, these should be there, the basics. And not only that, it also addresses the pre-marketing and post-marketing regulatory requirements. And one of the important requirements which is given in the guideline is that it is essential for the company to conduct phase 4 study, means pharmacovigilance study, with a minimum of 200 patients, within 2 years of getting approval for marketing. So once you get the approval for marketing, after that you'll have 2 years of time. Within that time, you have to do the post-marketing surveillance, means phase 4 study with the minimum of 200 patients, and that report is to be submitted. So students, you can ask me how many number of biosimilars are available in India. To my knowledge, now it is more than 95 biosimilar products are available in India. So in the EMA, that is European Union, you have only 64. In USA, we have only 19, but in India, so recent, the reports say that there are more than 95 biosimilar products are available in India. So I don't know how it has been, but these are approved and of course, which is by the different agencies, and it's available. So this is as per the report which I have taken. So dear attendees, so the next part is the development of biosimilars. Just I go a little bit quicker, it may be. So how the development of biosimilar by the different companies, and what are the the issues or quality, what it may be. Just I want to give you a little bit short, because the development of biosimilars is a very vast process, very vast process. Just I want to make it a short one. So biosimilarity to a reference product is established based on the totality of evidence approach. So you are comparing reference product with the biosimilar product. That means what? You are developing a biosimilar product and you are comparing with the reference product, and both should have similarity. That means you have to show similarity. That means you have to show the evidence. That evidence is known as totality of the evidence. So this is an approach through which the biosimilar is to be developed. How it is developed, we see. The bulk of the biosimilar development program is made of comprehensive analytical comparative testing. Just you are making a comparison. So just make a comparison. So that means comparative testing as a part of justification for demonstration of equivalence or similarity. So dear attendees, just I want to make you one thing sure, that you make a lot of analytical tests of a biosimilar product. So you justify that the biosimilar product is having very similar chemical structure to the reference product. So if the chemical structure is same, what may be the conclusion we can make it? The activity may be the same. So if the chemical structure is same, you can expect the similar activities, the same strategy, that is called totality of evidence, that is to be followed. That means you make a lot of analytical tests, you compare the reference with the biosimilar, and both should be same. If it is same, then you can say, or you can say what to call, it may be producing the same effectiveness, the same safety. So that is one. So because of that reason, you need not do much clinical work. That means the clinical part is very lean, very short. So if the clinical part is very small, then you will be saving lot of money in this case. So that is the reason the biosimilar products are available in the market at a cheaper rate. And even you produce a biosimilar which is having very similarity in structure with the reference product, there may be a uncertainties. So sometimes we call some extra groups, functional groups, or maybe the sheep, etc. So this is to be addressed. We call residual uncertainties. So everything which is not matching exactly, it's not identical, I already told you. So both the biosimilar products are not identical. So you need to tell what is extra or what is less. So that should be addressed through the analytical way. So I will talk about the production of the biological product. So I told you biosimilar product also a biological product only, and how this biological product is produced. So with this the picture, you can say this slide, I talk about the production of biological product or maybe the biosimilar product. First, you should know something is hiding, so that is reason just made a little small. So first you have to prepare a cell line. So you have to create a cell line. Of course, you should know the amino acid sequence. Then there are different process. Through the different process, that means you have to create a cell line. Through the cell line, you have to clone it. So you have to make a lot of clones. Out of those clones, you have to select only one. So in this case, one clone is selected. After selection of this clone is to be cultivated, for which you have to prepare a master cell blank. So cultivation or production, right? So you produce a master cell blank. So that means you have identified a suitable cell line. So this cell line is grown, or you're preparing a master cell blank. From this, you are preparing a working cell line. and from which you have to have a technology called expansion of the cell, and after that you have to produce the required product. That means a protein product which is produced after expansion of the cell. That means with the use of some bioreactor we call. So it's the required product is produced here. After production, you will have the product along with several other impurities that should be removed. So impurities removed. So for that, we need to have another step called filtration. Thereby, most of the impurities are removed. Then you have to go purify the product with the use of chromatographic technique. So you'll be getting the purified protein product. Then you compare the purified protein product with the reference standard. So here the reference product and here is the biosimilar product. So these should be same, right? So in this case, the examples of CQA means critical quality attributes. So some property, we check the property and compare with the reference product. So the property should be same. So this is checkpoint number 2. So earlier checkpoint number 1 is there. So you have selected the clone. The clone should be very similar to the reference product. So in this case also just we are comparing the reference product with the biosimilar product. That is also same. After that, once you produce product after purification, that may be formulated, fill, and you will get the finished product. So normally it is in the form of injection, I already told you. And again you check the critical quality attribute comparing with the reference standard, and that should have the similarity. Then you can say your biosimilar product is okay. So in between there are a lot of analytic tests to be done, lot of analytic tests to be done. That I am not going to highlight here. Maybe in the next slide I am going to tell you. So dear attendees, just you see the development of biosimilar. You have two triangles. The first triangle is here and the second triangle is here. So the first triangle is say the reference product development. So where you have to do little analytical work, more clinical, means preclinical work, then you have to do much more clinical phase 1 study, then lot of clinical phase studies, that means 2 and 3. This is the way how to develop a reference product, means initial or original product. But you see the second diagram, this is the development of biosimilar. In this case, you will be copying from the reference product. So just you know how the reference product is. So based on that, you have to make a similarity, means you produce a biosimilar product and you check whether the structure of the biosimilar product is same as that of the original product. That means comparison. For the comparison, what you have to do? You have to do lot of analytical work. Through the analytical work, you should justify that the molecular structure of the biosimilar and the molecular structure of the original drug are almost same. I am not telling you it should be identical. It is not possible to produce an identical biosimilar, but you can produce a biosimilar which is very similar to the original reference drug. So that means you have to do lot of analytical study. After that, you do preclinical studies, maybe in vitro study or in vivo study in the animals. There you can get much more confirmation of the activities or mechanism action, whatever may be, and that also can be compared with the reference drug. Then you do little pharmacokinetic studies or pharmacodynamic studies, and of course you do the clinical phase study. It is important just for the confirmation purpose only. Otherwise, when you complete your analytical studies, you can say that it's very similar, but these are the other steps that should be completed generally, because for the further confirmation of your developed biosimilar with the reference drug. So attendees, just I'm going to talk about the different stages of the biosimilars. First, I already told you the analytical. There are 4 stages. Of course, sometimes you can say 5 stages, including the phase 4 study. But here I'm going to give you only 4 stages. The biosimilar product development, 4 stages. First, you have to do the analytical studies. You compare reference product, the biosimilar product, and you make sure that the molecular structure are same, very similar, not exactly identical. Then you do the preclinical studies. So how? This is to confirm the biological function, for example, mode of action, between the biosimilar product and reference product. So here also, depending upon the conditions or the activities, you can use different models. Model sense, you can use the in vitro cell lines or different animal models, etc. So that what you want to the confirm. So that depending upon the the product, depending on product, what exactly you want to make it. So that can be used, that can be finished with the use of a suitable in vitro model or animal model. Then you move on to the clinical phase one. That means pharmacokinetic studies and pharmacodynamic studies in human. So this is to determine almost bioequivalence. No, bioequivalence exactly is not possible. However, just make a comparison of reference product with the biosimilar product and make sure that both are working in the body in a same way. Sometimes it is also not possible. Sometimes in the pharmacokinetic studies and pharmacodynamic studies, it is not possible to get the exact result. Why? Because the developed biosimilar product may have immunogenic. If that is the case, it is very, very difficult. So I will come this point maybe later on. So you have to do the pharmacokinetic studies and pharmacodynamic studies and make sure that both are having similar data. If that is the case, we can say it is passed. Now we will move on to the clinical study phase 3. In this, further confirmation of the safety and efficacy. And of course, that should be conducted in a sensitive patient population. Again, confirmation of the safety and efficacy. Again, we are comparing with the reference product and the biosimilar product only. So attendees, just this is a small picture. Again, tell the same thing only, the different stages. The first stage is the analysis, means so that's analytical test, then function test, maybe preclinical studies including animal data, then clinical studies, then post-marketing safety monitoring. So this is what I told you. This is a fifth stage is the post-marketing safety monitoring. This is a phase 4 study, pharmacovigilance study. Sometimes it's also required, right? So this is again a small repetition of the biosimilar development efficacy studies. How you will find the efficacy of a biosimilar product. So I already told you, just to compare the biological product, reference product with the biosimilar product. So if the structure is same, you can also say that the function may be same. That means two biological that are highly alike, similar, analytically, it almost inevitably have the same functions. So if the structure is same, you can say that activity also may be same. In that angle, you can say the efficacy study is to be conducted. So sensitivity and specificity of the modern analytical methods may discover appreciable analytical differences between the biologicals. So that means it's a biosimilar, means there may be small differences. So these differences may be identified with these of modern analytical methods. Of course, I tell you maybe in the next slide. So there are a lot of analytical techniques are available through which you can identify where the problem is, means where is the differences. And of course, generally the macromolecules, means protein molecules or biosimilar products or biological products, that should be characterized and compared at the different temperatures. So you keep different temperatures and you study the effect of different temperature of these molecules and compare. Then it is possible to exactly find out the differences between the reference product and the biosimilar product. So just I'll go a little bit faster, that how the analytical studies that helps in biosimilar drug development. So just comparison, I already told you, for the comparison purpose, you have to have certain the standards, or you can see what exactly you want to do it. So what you want to find it by doing the analytical test. So that's a question. So you are going to find the primary structure of the protein. Primary structure of the protein means you have to find the amino acid sequence. Then you are going to find the higher order structures, means it is secondary structure, tertiary structure, and coordinate structure of the protein. So first you have to find out the primary structure, then the higher order structure, generally we call secondary, tertiary, and coordinate structure, that should be identified. Second one. The third is post-translational modifications such as glycosylation or any other inclusion of different part or different functional group in the protein molecule. So we call it glycosylation, slylation, amidation, etc. So product related substances such as protein deamidation, oxidation. So these are the different modifications may be possible during the protein manufacturing. It may have some different structure. So these to be identified with the use of analytical techniques. That means with the use of analytical techniques, we are going to identify all these properties you can see. All right. So anything, whatever may be the analytical studies, you have to identify what? Some differences. So for which you need to have a lot of instruments, as I told you. So how to choose the instrument? Of course, you have to choose an instrument that will give a proper result. Okay, and that instrument that should be handled by some person. The person should be familiar with the instrument, and the instrument should give an efficient result. So there are a lot of the criteria for the selection of the instrument. So instrument means the operation of the instrument somebody has to know. It means they should be familiar with the instrument, and the instrument should give the efficient result. So keeping there are different criteria based on that the instrument techniques to be followed. So what are the instruments you can say? Generally we call LCMS. So generally, so for any kind LCMS is good one, and of course maybe the combination of LCMS means HPLC and LCMS. We call HPLC and MS. So that may be combined. Sometimes LCMS, MS, of course. In this case, capillary electrophoresis may also be possible. And generally, it is expected that the company should have some software program to identify the different functions. So automated data to evaluate the protein identity, sequence variants, glycosylation profile, post-translational modification, impurities. For these, we have to have an instrument definitely. Along with that, there should be a software so that it will be easily and automatically find out all this. So that is good for the characterization. So attendees, to determine the higher order protein structure, we need to have some more instruments, maybe very costly instruments you can see. Circular dichromation. So this is for the second structural characterization. Then FTIR, that is for the secondary structural characterization. We have DSC, that means differential scanning calorimetry, size exclusion chromatography with multi-angle static light scattering. This is for the quaternary structure. So it may be a little difficult sometimes to the students, especially who are studying at lower level, maybe first year, second year, maybe sometimes third year also may be having some difficulty in understanding all this. These are different instruments which are supposed to be used to determine the structure of the protein. So in case of circular dichorism, we can find out the structure and interaction of the proteins, the metals and other molecules. DSC is differential scanning, calorimetry. So that will be finding the conformational. Conformation means shape of the molecule, shape of the protein molecule in fact. So and the thermal stability of the biopolymers protein molecule. So there are different instruments to be used for different purposes, and sometimes we have to use all together for the characterization of the proteins. So here is another table again that will tell you how the different structure is to be characterized, what you are going to characterize, and what are the methods to be used. So I'm not going to deep into this because already I told you. So some instruments which is given as a picture, and this is the diagram which is already given. So LCMS instrument here, and DSC picture, just means it's a diagram, and of course circular dichorism diagram which is available here. So these are different instruments supposed to be used. So what are maybe the instrument you are using, and finally you'll come to some conclusion, right? So there are four type of conclusions possible with the use of the comprehensive analytical studies. It can arrive into four assessment. The first assessment, it is not similar. The second is similar. Third is highly similar, and fourth is highly similar with the fingerprint like similarities. That means four assessment. So if it's not similar, it is not biosimilar. Just we can leave it. So any assessment that comes under the category of not similar, that suggests that the further development through the biosimilar pathway may not be appropriate. So you should not do it. Just stop it, and you have to produce another product. Means you change your route or whatever may be, the product is not similar, you have to restart again. But if the conclusion is the similar or highly similar with the fingerprint like similarity, you can choose that product for further development. That means you are going to develop a biosimilar product once you get the result as similar, minimum similar, right? So then you can go for the next steps. The next step is the so the clinical studies or non-clinical studies, etc. So just a few things just I want to highlight here. Non-clinical study with the use of in vitro cells. So what you have to do here? So you can test the protein on in vitro cell lines, but provided that you make sure that the whatever test you are performing in the cell lines, so those tests should be giving answer in the in vivo test. That means the test should include all functionalities known to be active in vivo. So just like that, you should not take any cell lines. You have to choose a cell line and you do the test, and that test should be exploited even in vivo. So that is one. So the difference between biosimilar and differential product, that should be fall within the predefined limits. So I told you the exact copy is not possible, so that you cannot expect the exact similarity. It is not possible. So that if some predefined limit, if it is available, and your similar biosimilar product should come within that predefined limit, maybe plus or minus 5%, 10%, something like. Then what is the need of these human pharmacokinetic studies? So this serves several purposes. By comparing the pharmacokinetic data of biosimilar product and the reference product, it gives some important information that it's a complement to the data from analytical studies, and in confirming the likeness of the composition of the two products, particularly with regards to glycosylation and other post-translational changes. Just comparing reference product with the biosimilar product. So both are same. What you can say? The structure is same. If activity is same, you can say the structure is same. So that is a likeness. So if the structure is same, you can expect the activity is same. So that is the only one good information you'll get. So it is also glycosylation. So there are different shape of the protein when you produce a biological product. So sometimes it is very difficult to do the characterization in the analytical studies. But when you do the pharmacokinetic studies or clinical studies, we can say, then you'll be getting the same kind of activity. Then you can see the all glycosylation or post translation changes, everything is okay, means similarities. The next is this constitutes a sensitive method to support the likeness of a primary component of clinical effectiveness. So pharmacokinetic study also say that this effectiveness of that biosimilar product. The third important thing is that this provide an evidence of any excess immunogenicity of a biosimilar product. So dear attendees, immunogenicity is a very the big topic, and you have to produce evidence that the product will not produce any immunogenicity. Means biosimilar product should not produce any immunogenicity. So why? Because when you take a biological product, a protein product into our body, so body also produces a lot of antigens. So antigen means it's a protein which you are taking, and antibodies are produced because of the this protein product. So because of that reason, the antibodies go and attach to the protein product and make it not effective. That means useless. So that is called immunogenicity of the product. So we make sure that the immunogenicity of that product, means the protein product, is very less. If it is less, it will be giving activity in our body. If it is more, it will not give you any activity, because antibodies go and bind to the active sites of the protein product, means biosimilar product, and it will not give any biological activity in our body. Sorry. So their attendees make sure that the protein product should not show excess immunogenicity activity. So this can be seen with the use of pharmacokinetic study. So pharmacokinetic data which also tell us the immunogenicity of the biosimilar product. Okay, so evidence it says, and also it also records the adverse events. So pharmacokinetic studies serve several purposes. It must be done. And the pharmacodynamic studies. So this is a study when you give little quantity of the dose to the animal or maybe the human body. So what is the response? If we give more quantity, means if you increase the dose, what will be the the response of the body. So that is known as pharmacodynamic studies. So sometimes the markers may not be available, and sometimes this is to confirm the biosimilarity. So somehow this is to be done, and these are the studies generally to be carried out. This is to check the relationship between the drug concentration and the pharmacological effect. That's what, if you increase the dose, what will happen to the body. So that is to be identified. So students, I already told you the immunogenicity study that should be carried out through the pharmacokinetic study. So then you must do the clinical studies. So that recur generally for the biosimilar, but in a very, very small manner. So that is the reason you will be saving a lot of money for clinical studies when compared with the development of the original biological drug. Sometimes the people say these are unnecessary hindrance. So it's unnecessary, just we have to prove again. But however, it should be done. So all the regulatory agency, they ask the clinical study of this, and after that only the regulatory agency approve the product to be marketed. So again, the biosimilar development process given in this single slide. There are a lot of things are available, and once you produce an evidence, what is evidence? It's a totality of the evidence. When you produce such kind of evidence, after that you submit your the findings to the regulatory authorities, then the regulatory authorities will give you the approval. So after that, you can market your biosimilar product in the market. Okay, attendees, we are going to coming at the end, maybe another few slides only. Just I go a little faster. What are the challenges? So is it possible to produce or market the biosimilar easily? Or what are the problems in between we will have? Of course, I told you there are lot of challenges are available. You have to read completely before producing a biosimilar product. You have to read, you have to have an in-depth knowledge. Or you are working in a biosimilar product development as a a part of the team, you must know everything. You can say each and every part related to the biosimilar. So that we call as challenges. So demand for biological drugs are increasing. So generally increasing means we have to have a good opportunity. So when compared with the small molecules, we have a lot of opportunities. That is not a challenge, but you have to have a lot of challenges. I told you the biosimilar landscape complications associated with the regulatory approval, manufacturing problems, bioanalytical the characterization and marketing concern. So these are the problem. So we have to know what there are a lot of trouble with the regulatory approval. So we have to completely know how the biosimilars are get approved. That means how the approval is possible. You have to have an in-depth knowledge in this. And manufacturing, of course, I told you, you do a lot of manufacturing, the strategy, and after that you will get a product which is not biosimilar. Then what of the work you have done is complete is going to be wasted. So that means you have to have very depth knowledge in the manufacturing, and you have to have a big, analytical lab, bioanalytical lab, where you have to have a lot of people working for that particular lab, and they have to have very good knowledge in the characterization of the biosimilar product. Of course, the marketing, such a very, very big, without marketing it's not possible to get the money. So that is also very, very, the important field. So these are different challenges, right? So one challenge that is the development that may be solved, may be minimized by outsourcing. Means outsourcing could lead to the cost effective development while minimizing some challenges. So it is possible outsourcing. So you just give the money to the biosimilar product development company. They develop for you so that you can minimize the cost effective development, means the challenges. And once you have the in-depth knowledge, you can overcome some of the challenges. That means you have to have knowledge of each region, whether biological product or maybe biosimilar products work in that region or not. Early strategy planning. So this is also very, very important. So along with the regulatory authorities, you have to have a meeting or means continuous touch with that, and effective communication with the regulatory agencies. So what type of products needed? What are the problems already faced by the different regulatory agencies or the companies? And you have to have a good communication with the regulatory agencies. They will guide you. So these are different challenges. And successful development and commercialization of the biosimilars require business strategies that integrate appropriate clinical design and regulatory compliance. That means ultimately you have to sell your biosimilar product in the market, so that you have to have a business strategies. So that I not tell you much about this, but this is also a challenge. So there are a lot of other challenges. Just I want to summarize here. So it's difficult to produce two different manufacture same biologicals that is identical to each other because of the variability of post-translation modifications such as glycosylation, phosphorylation, carboxylation. So these are different, post-translational modification. Why that comes? Because of the degradation, aggregation, deamination, in view of differences in the conformational changes as well as in the immunogenicity. So please make a note. During the manufacturing, there are a lot of steps are available. So during those steps, there is a different changes or modification occurs because of glycolation, etc. Why this occurs? Because of degradation, because of aggregation. So the product going to aggregate, degrade, or there may be deamination is possible. Why? Because of these reasons, there will be a change in the shape of the molecule, and because of the change of the molecule, there may be a difference in the immunogenicity. So students or the attendees make a note immunogenicity. That's why I told you the product should show less immunogenicity, then only it can be marketed as a drug, right? Otherwise it is not possible. So these are different challenges that should be optimized. Means each and every step during the manufacturing that should be optimized to get a product that will be showing less immunogenicity ultimately. Right. So for which you have to have a cell culture. You have to identify a suitable cell culture, and cell culture storage. You see how much time that can be stored, at what temperature that can be stored. So these are different challenges one person will be having, so the company will be having. Small modification also that may lead to some changes. What are the changes? The changes may be on the pharmacokinetic parameter, or it may be getting the more immunogenic problem. There may be changes in the post-translation modification. There may be changes in the shape of the molecule. Because of that reason, there may be a stability problem. There may be a safety problem. There may be an efficacy problem. That means each and every step during the manufacturing or during the optimization of the drug development, that should be optimized. Again, there are other challenges also. So I already told you biosimilar product development, that is based on the reference product only. You have to have a reference product and you have to compare it. So if reference product is not available to you, what to do? You are comparing reference product, and if the reference product is not available, it's very, very difficult. You cannot. So this is also a major problem. So there are companies, they produce reference product. They will not give you the reference product. So they implement a self-imposed restricted distribution system. They don't give you. So if they don't give you, then it is not possible to produce a biosimilar product. That means the brand name company generally able to prevent or delay the biosimilar product from a company. So that record testing generally this is also possible. So a company also may be preventing or delaying another company biosimilar product development because they will not give you the reference product. So this is also another challenge. So there are other challenges also, the patient and prescriber education. So biosimilar products available in the market, say for an example, and the patient must know that this particular biosimilar product is available that is useful for his disease. And similarly, the prescriber means doctor, they also should know. Then only the biosimilar that should be used. Otherwise, it cannot be. That means the physician, pharmacist, the patients all need to be enlightened and subsequently convinced of the benefits of switching to the biosimilar. So everybody, including pharmacists, nurses, patients, or we can say healthcare team along with the patients, they should be educated with the availability of the biosimilar products, the benefits of the biosimilar products, so that they can easily switch to the biosimilars from the conventional drug. This is also another challenge. Interchangeability question. I already told you the interchangeable product. So sometimes we need to have only pharmacist. Just he will interchange or substitute the product that we call interchangeable product. So here also sometimes the doctor intervention may be there. So you need to have, this is also a kind of challenge, I can say, another challenge, because the pharmacist himself, it is not possible to substitute. And in case of rare disease, it is not possible to produce, the biosimilar product because of some troubles. Why? The trouble is difficult to obtain large enough and non-heterogeneous population for phase 1 study and phase 3 clinical trials. Because the disease people are very less money, then how it is possible to produce and how it is possible to test on those people is difficult. So we call rare disease. So to tell you about the rare disease, acute lymphoblastic leukemia, so it's a cancer, it's a melanoma, so cancer. For this disease, and we call rare disease, and the drug is called orphan drugs, for which it is very difficult to produce the biosimilars. So that is also another challenge that we are facing in the biosimilar development. So, dear attendees, I talk about the safety of biosimilars. I told you the biosimilars are safe, but however, just some information. Because biological drugs are administered parentally, devices are required. So devices are required in this case. So the biosimilar products generally is to be given in the form of injection or infusion. It is not to be taken orally. If it's taken orally, it is not effective. So it should be given in the form of injection only or infusion. So in that case, there are devices required. So the design of the device for the biosimilar product, it will have some, the function will be different, the appearance will be different, and also patentable. So the the pharmacist, or we can say the doctor, nurses, they must know about these biosimilar device design, its function, appearance. They should know how to handle it. Then only it's possible. So this also comes under safety of the biosimilars. And the pharmacovigilance, I already told you, it is important. Sometimes, the biosimilar product will have certain, uncertainties, certain extra functional group. So that functional group may produce some kind of activities, may not be immediately, maybe in the later stage. So that is the reason the pharmacovigilance study is to be carried out. And of course, the safety is to be considered on a drug by drug basis. So every time that should be checked. So what is the feature perspective? So what is the feature of this biosimilar? Of course, you have a lot of chances of biosimilars in the future. The biosimilar product in the future will be more complex. Even now also, it's a more complex only, but even compared with the present biosimilar product, the future product will be multi. Just I want to tell you, glycosylation means the attachment of the sugar group into the protein. So similar, so that's called modification. So a lot of sugar may be attached, a lot of lipid may be attached, something like. So that becomes very complicated one. In the future, that may be available. So you should be ready to handle such kind of biosimilar product in the future. So there are many companies, they also enter the market, produce biosimilar product, and they make fast progress. That means in the future, there are a lot of biosimilar products going to be available. So that is the reason the key stakeholders like prescribers, means we call doctors, pharmacists, nurses, patients, and any person who will be involving in the healthcare, that means healthcare team, including the company, manufacturer, will be facing a lot of challenges. The education is required and handling, etc. So these are the different challenges also, and those challenges to be addressed also. That is future perspective. The acceptance of biosimilars by the general public and their ample use in healthcare is also another challenge. Of course, it's a challenge also, and of course, in the future this is going to be happen. So a lot of people will be going towards the biosimilar product, and for which you need to have education. So what will happen to the industry? So in the future, many industries they focus on the biosimilar product. It is not only in the US and Europe, but that is also throughout the world. Any country they may be focusing on the biosimilar product in the future. Because of that reason, there will be competition. There will be a approval. So several approval of the biosimilar products, rapid introduction in the market. So because many biosimilars is going to come in the future, so the market is to be evolved to adapt. So market has to change itself to agree or accept the change means availability of the biosimilar products. So this is a market that will remain in focus in this billion-dollar industry with the possibility of changing industry forever. See, because of that reason, the industry is going to be changed in the future. So I'll be talking about the administration. Very small only. I already told you the patients receive biological by injection only under the skin. So it's called subcutaneous injection or by intravenous infusion injection, because that cannot be taken orally. If it's taken orally, what happens? The process of digestion breaks the biological product and make it ineffective. So you should not take the biological product orally. So that's a very, very important one. And here I show you the different devices with the different companies and operation, etc. So all this education is must for us. So dear attendees, I think you'll be very happy just because I'm concluding my the webinar. So in the future, there are many biosimilar products going to be available in the market, so that the pharmacist, it's our role that you have to handle the biosimilars in a proper manner, including the storage and what conditions, how long it can be stored, at what temperature stored. So these knowledge you must have, and you have to have a very good education with respect to the biosimilars. Like yourself, you have to educate about the biosimilars, and you have to educate the patients and healthcare team, including doctors, nurses, or any other person who involved with the healthcare team. You should educate them. What you have to educate? You have to educate, of course, every aspect of the biosimilar product, including the device. You can also suggest the doses or any other relevant information related to the biosimilars to the healthcare team. And not only that, your big role also and in the pharmacovigilance studies. That means you have to report the adverse reaction which is produced by the biosimilar product. So attendees, so just with this, I'm concluding my webinar. Maybe I have taken more time, but I think it is fruitful to you. So those students who are eager to go for a biosimilar product development or biological, biopharmaceutical company, I think these are the basic, information that will be suitable to you. So with this, I thank every one of you, all the attendees, students, maybe some of the staffs also. You are the principal of the college also. So with this, just I thank all of you. Any questions?
Thank you, sir.
Thank you, sir.
Forum
Thank
is
you,
open
sir.
for question.
Thank you. Thank you so much. Any questions? Any questions from the
Sir, good evening, sir.
staff or students?
Sir, good evening, sir.
Good evening.
Sir, hello, sir. Sir, this is
Please.
Arjun, sir. Sir,
Arjun,
actually, I have
please.
two questions, sir.
Please.
First one is, if there is a innovation in biosimilar products, they can go for patent, na, sir.
Yes. That becomes a new biological product.
Yeah, sure, sir. Actually, if they got patent, the cost of the product is going to high, na, sir.
Yeah, you have to patent it because it's a new biological product. You should patent it.
Okay, sir. In that case, sir, the biosimilar product cost is going to high, na, sir.
I'm not getting it. The cost of what?
Sir.
Yes.
Hello, sir.
There's some disturbances. I tell you, the cost is going to be high. Yes, your question is right only. Because it becomes a new biological product. It's not biological product. It becomes new biological product.
Okay, sir. Okay, sir.
It is some disturbances.
இப்போ நம்ம பொருள் வாங்குறது வச்சுக்கோங்க, இப்போ பெயிண்டிங் மட்டும் நமக்கு தனி அமௌண்ட் வருமா?
Hello. I feel some disturbance. Please down.
பெயிண்ட்டுக்கு மட்டும் தனி அமௌண்ட் வருமா? வேற தனி சார்ஜ் கேட்பாங்களா பெயிண்ட் அடிக்கிறதுக்கு? நம்ம அது கேட்குறேன். ஆமா ஆமா,
Any question?
சொன்னாங்க. Hello, sir. Hello, sir.
Hello. Yes.
Yeah.
Can you hear me?
Sir. Yeah,
Please.
I'm hearing you, sir. Actually, I have one more question, sir.
Please.
Is there any difference between generic drug and biosimilar products, sir? Is there any difference? If there is a difference, can you elaborate the difference, sir?
Yeah. Just you want to make sure that the generic that is applicable only for the small drugs, small molecules.
Okay, sir.
But in case of biosimilar drugs, there is no question of generics. The biosimilars, they're supposed to be generics, but exact copy is not possible. That is the reason we call biosimilar.
Okay, sir. Sir,
This word should not be used. That's what.
Yeah, sure. Sir, approximately how long it will take to produce a biosimilar product, sir? Approximately how long it will take, sir?
Again, I tell you, this depends upon the company and how many people are working, what exactly the complexity of the protein, etc. But generally, it may be around 6 years up to 10 years, something like. But
Sure, sir.
nowadays, there are so many instruments are available. Maybe around 5 years, 6 years, they are going to develop.
Okay, sir. In case if there is a patent for reference product,
Okay.
if I want to produce a biosimilar product, after the patent is expired only, I have to start the production, sir? Or
Yes. Many companies they do the same thing only. Many companies, what they do, they start producing biosimilar product and just they will be waiting for the end of the patent of a reference product. Then immediately they patent it. I mean, so they release it in the market.
Thank you, sir.
Welcome, please. Any questions from the student side? I'll be very happy to have questions from the student side.
So feel free to ask the questions.
Please.
Any other questions? Students, any other questions?
Okay, sir. Okay. Professor Dr. Mahadev Anjan, sir.
Yeah, please.
This is Dr. Venkatesh, sir.
Principal.
You had a wonderful and informative presentation, sir. Your presentation went on very well.
Thank you.
Thank you, sir. On behalf of our management of AKCT, hereby express my wholehearted thanks to you and your management for giving such a informative and elaborated presentation. So, can you hear?
Yeah, please. Thank you so much.
Wonderful presentation. So some seeds of intelligence in our students. I hope this interaction presentation will motivate the students and encourage them in active learning in the forthcoming days, sir.
Definitely, sure.
Yes, sir. Thank you, sir. Sir, one more request, sir. Please let me know in future, sir. I would like to plan your native Tamil Nadu, sir.
Okay.
Please let me know, sir. We will arrange international seminar, sir. We will invite
Please,
you.
no problem. But I may be coming the next year only. Maybe in the month of June only. I may be planning.
Whenever, sir. Whenever
okay.
would like.
Yeah,
Sir.
please. It's my pleasure to join you.
Yes, sir. Sure, sir. Along with your family.
Please. Happy.
Sir, thank you, sir.
Happy to come to you, please.
Thank you, sir. Thank you, sir. Take care, sir. Take care.
Thank you so much. Should I leave now?
Well, yes, sir.
Leave?
Yeah. Leave.
So thank you so much to the principal, and also thank to Dr. Aritinarishwaran, who was in constant touch with me. And please, you also keep in touch with me even here after the now also.
Yes, sir. Same also.
Thank you
Let's
so
have a
much.
good collaboration.
Thank you for your opportunity. Thank you so much. Bye.
Good collaboration for future, sir.
Please, thank you. It's my
Okay,
pleasure.
thank you.
Welcome, please. Welcome. So bye.
Bye.